a very great pleasure to welcome you here tonight, both in person and online. And it's a particular pleasure to welcome our speaker, Professor Raina MacIntyre, known, I think, to many of us during those long COVID months and even years. I think we've almost forgotten some of the grimmer scenes of those mass graves in New York, or those lethal, disorganised hospital wards in Italy. But we shouldn't forget that here in Australia, our government's put forward a rather impressive effort, despite a lot of opposition from some parts of politics. And not only that, but we had community leaders like Raina MacIntyre, who explained things to us, put forward the case for taking the precautions we had to take, and to whom we owe a great deal. Professor MacIntyre, who's Professor of Global Biosecurity at the Kirby Institute at the University of New South Wales, has recently put out a book called Dark Winter, in which she explores the background not only to COVID, but to a range of epidemics, pandemics, both in the past and, who knows, in the future, and explores some of the less discussed and perhaps more worrying ways in which some of these have originated, things like laboratory leaks and illegitimate political activity, to put it politely. But I'm not going to say any more because we've got an expert here, so it's over to Professor MacIntyre.
Thanks so much, and good evening, everyone. Just like to acknowledge the Gadigal people of the Eora Nation and pay my respects to elders past and present and any Aboriginal or Torres Strait Islander people here today. So I had a really nice chat with the interns just before this, and I might start by telling you something that's been in the news in the last few days, which you may have even seen, which touches on US-Chinese relations, which one of the interns is actually doing their honours thesis on. There was an illegal lab discovered in Reedley, California, which is near Fresno. And it was basically a Chinese-owned lab which had belonging to this company called Prestige Biotech, which had been formed after the parent company had gone bankrupt, a company called Universal Meditech, which had been making COVID tests and a few other things through fires in their previous laboratories, one in 2020, one in 2022. The FDA recalled their COVID tests in 2022 because they weren't produced according to the regulations. And then just an ordinary council worker was walking past this old warehouse and noticed a hose sticking out of the wall. Now, when police go looking for drug labs and meth labs, that's a pretty indicative sign, seeing a hose sticking out of the wall. So that raised the alarm, and they actually reported it to the FBI at the time in December 2022. Nothing much happened, and then they got a warrant 3 months later to go in and inspect the premises. So March 2023, they went in and found, nearly 1,000 mice that had been genetically engineered to transmit COVID, and a range of pathogens were identified by the CDC, including HIV, SARS-CoV-2, I think herpes virus, rubella, and a range of other pathogens, and lots of freezers and fridges and biological samples, chemicals of all kinds. And maybe it was just a company that went bankrupt because they had shoddy practices and they moved all their stock to an illegal warehouse where they shouldn't have been keeping it. Maybe there was nothing more to it than that, but this was a lab with connections to China, 35 kilometres from a military base, a naval base, and nobody seemed to be on the ball to the implications of this lab. Now, supposing for any particular country, supposing a hostile nation comes in and sets up a company saying we're making cars, we're making automobiles. That's the cover story. But they're actually making a nuclear bomb, right? So it's a Trojan horse type approach, which is much easier to do with biological agents than it is with nuclear agents. So, and I'm pretty sure if an American company had tried to set up something like this in China, they would have had a lot more difficulty than a Chinese company did setting it up in the US. Now, you know, I'm not making any sort of partisan comments about China or the US, but in terms of biological threats, it's a huge gap in the awareness of law enforcement, military and intelligence sectors, and you just have to look at the history of the sort of debate that's gone on about the origin of COVID. I mean, I've been following it, and lately there's been a lot of information that's been released under freedom of information in the US, and there's been a congressional hearing where various people, including scientists, have been interviewed, and it's looking pretty likely that it was a lab leak from the lab in Wuhan, which had also received funding from the US NIH. So that was probably one of the reasons why there was kind of quite a concerted effort to put out a narrative that it had originated in the wet market. Anyway, so in my research over the years, you know, I've done a fair bit of work on, but I do research on lots of different aspects of infectious diseases, but one of the areas has been biological warfare and bioterrorism, and prevention of it and detection of it. And I think it's one of the reasons in 2020, you know, New South Press contacted me and said, would you like to write a book about the pandemic? And I said, not really, you know, I'm really busy. Why don't you get back to me next year? So they contacted me again in 2021, and I said, okay, I'll write this book. And so I put it together, and it kind of crystallizes stuff that I teach in a course we do on bioterrorism and health intelligence, and on a lot of the stuff in my research. And the reason I wrote it is that, you know, I'd actually written a novel before that to try and get the message out that biological technology that's available today is absolutely mind-blowing. You know, most people who are not working in the field have no idea the kind of technology that's available today, and it is much more accessible, it's much cheaper, and there are published open source methods for how to either do gain-of-function research, which is to take a virus and pass it through an animal species over and over again until it adapts to that species. So we have humanized mice, for example, right, which were being used in the lab in Wuhan, the coronavirus lab. That's a mouse that's been genetically engineered to have the same respiratory tract features as a human being. So it's an analogue of humans. You can't really, we'd hope nobody's experimenting on humans, but the next best thing is a humanized mouse. So, and you'd have to wonder, you know, I wondered the lab in Reedley, California, you know, were they humanized mice? You've got that gain-of-function research, you've got synthetic biology, which is where you can make a virus from scratch in a lab. And the first virus that was made synthetically was a poliovirus around 2000. So it's technology that's been around for a while. But that's an RNA virus and quite a small genome. People have often thought smallpox, that the likelihood of smallpox re-emerging is very low because it's only held in 2 known locations in the world. One is at the CDC in the US, and the other is in the Vector lab in Russia. And it'd be very hard to break into either of those labs and steal it. But in 2017, some Canadian scientists made a very closely related orthopox virus, an extinct orthopox virus, in a lab from scratch. Wasn't that expensive, and it wasn't like a top-of-the-range whiz-bang lab. It was, you know, middle-of-the-range lab. and they published their methods on how to do this. And no one knew about it till after the fact, right? If intelligence agencies are looking for this kind of thing, none of them spotted it. So it caused a bit of a furor, because what it showed was that actually someone could make smallpox in a lab. You don't have to break into Vector or CDC to steal it. You can make it. And you know, there's been a lot of controversy around gain-of-function research, which is taking a virus that occurs in nature and conferring on it properties that are unnatural, that make it more dangerous for human beings. So either making it more transmissible, more lethal, or drug-resistant, vaccine-resistant, etc. And then in around 2002, I think, some Australian scientists who were trying to solve the mouse plague. You know, remember the footage we saw on TV of all the mice around those houses in the country? They were trying to genetically engineer the mice to make them sterile, infertile, right, by creating antibodies to their eggs. And they used mousepox, which is another closely related virus to smallpox, which only causes asymptomatic or very mild infection in the mice, but they engineered it to help generate these antibodies to the mouse's eggs to make them infertile. And it had two unexpected side effects. So they were trying to do something good, right? But it had these dangerous, unexpected side effects. And one was the mice all died. So they made it universally fatal, the virus. And the second was that it was resistant to the smallpox vaccine. So the smallpox vaccines work against any orthopox virus. You know, you remember last year when we had that unprecedented monkeypox epidemic, everyone was scrambling to get the smallpox vaccines that had been, you know, originally developed for smallpox, the third generation vaccines, but were used for monkeypox. So not only can you make smallpox in a lab, you can also make it resistant to vaccines and probably more fatal than it was in nature, which was about 30% death rate compared to less than 2% for SARS-CoV-2. So this is the kind of, and the problem with the biological technology that I've written about in the book is that the methods are available to anyone. It's not like nuclear technology, which is much more difficult for any random actor to pick up and use. It's very accessible. You can buy DNA sequences online, you can buy a lab-in-a-box kit online, you can set up a nefarious lab in the community and nobody would know about it because no one's looking for it, just like that lab in Reedley. I was invited to join a group called Biosafety Now in the US, and that's a group of people who've come together around these concerns, and I was happy to join them because I sort of felt like a bit of a lone voice, but I realised there's other people who are concerned about this. And the debate around what's called dual-use technology, which is technology that is intended for good but can also be used for harm, and it can apply to anything, to AI, robotics, biological technology, cyber technology. But the problem with dual-use biological technology is it can cause a pandemic. And in other research that I published in 2015, I calculated that the risk of an unnatural or man-made pandemic is orders of magnitude higher than a natural pandemic. So in my view, that is actually an existential threat that we're facing, because it's kind of, the sky's the limit in terms of what's out there. At the same time, the advances in genetic engineering through tools like CRISPR-Cas9, which started becoming available in 2012, about 10 years ago, means that you can engineer anything with DNA, plants, viruses, animals, human beings. And in the UK and Sweden, experiments started on engineering human embryos in 2016. Then in 2018, I think, a Chinese scientist took it a step further and presented his research at a conference where not only had he engineered human embryos, they actually were carried to term, and these two babies were born who'd been genetically engineered for certain characteristics. It caused a furor around the world, and China sort of, it looked like the guy was, put under house arrest, and there were consequences, etc. But very soon after that, everyone was scrambling to get on the bandwagon. The WHO put out guidelines on human germline editing, and then, other superpowers began publishing guidelines, and the UK is doing experiments on engineering human beings to produce super soldiers, as is the US. You can Google it if you don't believe me. There's an article in The Conversation, and probably China's doing it as well, and other superpowers. In the UK, it's not even accessible through freedom of information, so it's like totally secretive. No one in the public can ever find out what's going on. So when you think about it from a geopolitical stance, you can imagine that a hostile state may find a way of introducing something to a population, a target population, that alters the genome of the people, and then can attack them with a virus that's been engineered to hit a more susceptible population. So it's like a double whammy. It's kind of really quite the capacity. I've got a colleague who was full-time in the army, but he's now an academic and a reservist and an expert in CBRN, chemical, biological, radiation, nuclear explosives, CBRNE. And he thinks it's the arms race, is engineering of human beings. It's the current future arms race. So these are the kind of things I try to, concepts I try to introduce in the book, and it's written in very lay terms, so it's not technical, and it's easy to understand. And I kind of draw some parallels with climate change, where with climate change there's obviously a lot of vested interests that prevent governments from taking action, but people being aware of it and actually feeling the consequences of climate change, whether it's floods or fires or smoke from bushfires, people know it's happening because it's affecting their lives or the lives of people they love. So I think that awareness can help drive change. And so, my goal in writing that book was really to raise awareness about the risks posed by the biological technology available today. And I don't know, maybe Ian has some thoughts after reading some of the chapters, or questions he'd like to ask me.
All of the chapters. It's a terrific book. Well, as I sent you in conversation, one of the shocking things to me was the nastiness that you've been receiving for daring to raise questions like the real origins, whether of COVID, but particularly the laboratory leak in Wuhan, or indeed going back things like anthrax, and then going forward, what's it going to be now? What's going on in laboratories now? So, looking at your book, let me think what I'd like you to talk about a bit more, because there's so much in it. I do recommend it to everyone. Like that. Your very final chapters about a biological winter. I think it would be very nice to hear a bit more about those.
Yeah, so there's a real imbalance in the regulation of the kind of technology I've talked about in that it's any regulation that there has been or there is has been heavily driven by protecting the interests of scientists, and very little engagement from the community. There's not really been a community voice at the table. We actually did some research and published a paper looking at community awareness of this kind of research, and it's extremely low. Most people have no idea, and when informed about it, most people find it unacceptable. So the community ultimately is the stakeholder, whether it's from a lab accident or whether it's from new drugs or vaccines that are developed. It's the community that's the most important stakeholder. So communities should, be informed and engaged, and I think that's the way forward. I don't believe any solutions are going to come from the scientific community, because there's too much conflict of interest, vested interests. You know, it's a huge industry. It's a bit like the military-industrial complex. It's a huge industry, pandemics, and I think the solution. I do believe it's an existential threat, and I think the solution is from people being empowered with knowledge and being able to have a voice. It doesn't mean banning that kind of research, but better regulation. It really has to be global, because an experiment done in one country can result in a lab leak that affects people in another country that didn't consent to that research nor were informed about it. And that, to me, is a new issue in terms of the ethics of research, you know. So research ethics has developed out of the Nuremberg doctors' trials and the Declaration of Helsinki, and you know, the principle is around individual patients as subjects in medical research. So the whole focus is on an individual having to be fully informed of what the risks are to any research that they're participating in, and consenting to that research, and also of not doing any harm. There's got to be, you know, you can't do research that causes harm to human beings. But when you're talking about pandemic pathogens, a lot of the research is actually happening in animals. You know, influenza gain of function research is done in ferrets, which are a good model for humans. Coronavirus research is done in humanized mice. So those kind of research projects go through animal research committees, which don't consider, you know, is someone in another country going to be harmed by this? And I actually think that kind of research should be registered publicly, like we register clinical trials. So when I started doing research, there was no requirement to register a clinical trial. Around 2007, 2008, that changed, where you couldn't do a clinical trial and get it. You couldn't get funding pretty much from a reputable funding agency, and you couldn't publish the results of a clinical trial in any reputable journal unless it was registered. And the reason that happened because the pharmaceutical industry was often starting trials on promising drugs and then just covering up the results because the results were not favourable. So it was to make sure that everything was in the public and, so that things couldn't be covered up in terms of unfavourable drugs. And that was a huge culture shift in medical research because it required everyone to come together and enforce it. So it required medical journals to refuse to publish anything that wasn't registered, a trial that wasn't registered. Prior to that, you could publish a trial that wasn't registered. It required funding agencies like NHMRC to not fund a trial that wasn't registered. And it required journals to also come on board. Yeah, question? But yeah, while we're waiting for the microphone to get down there, that cultural shift did happen, and it was a massive shift.
Chris Skinner, a counsellor, you said that this is an existential threat, which one might think is more immediate than climate change. What is the World Health Organization doing? Is it showing sufficient leadership?
They have got a committee called SAGO, S-A-G-O, which looks into the origin of new epidemics. So they're specifically tasked with trying to identify whether they're natural or unnatural origin pandemics or epidemics. They've got another committee that's been put together, I think, it's got WHO backing, but it's called the Pathogen Project. So there's a bunch of people
look, that's not really what I'm talking about. You just said a moment ago the community is unaware.
Yeah.
Well, what is the World Health Organization doing to inform them?
Probably not enough. They have taken some steps to try and, but they're sort of expert committees rather than going out and informing people. It really is a bit like, you know, there's very powerful vested interests at play. So it is that step of informing the community. And as we saw with the WHO, you know, if you remember back in March 2020, our Prime Minister declared a pandemic before the WHO did. It was bleedingly obvious to everyone it was a pandemic, right? But WHO is holding back for political reasons because they have to manage the stakeholder relationships with donor states, and China is a major donor state to WHO, as is the US. So there's political interference that comes into that kind of hinders the ability of WHO to sort of address certain things.
Okay. Can you say a little more about gain of function research? It's a constant theme in your book.
Yeah. So gain of function research first kind of came to prominence in 2011 when two groups of scientists in the US independently wanted to publish their research, which showed the method for how you could take an avian flu virus. So avian influenza is only contagious between birds. Birds have different receptors in their respiratory tract to humans. So if, occasionally humans do get infected with avian flu, but it's not contagious between humans because of that receptor difference. So if, a pandemic could arise if the receptors in if the avian flu virus gains the affinity to the human upper respiratory tract, right? So these two groups had done exactly that. They had engineered avian influenza viruses, which, these are pretty deadly in humans when humans get them, sort of 50% fatality rate. They'd engineered them to be adapted to the human respiratory tract and potentially cause a human pandemic. So it caused controversy globally. The only country at the time that had any mechanism to even deal with this was the US. They had something called the National Science Advisory Board on Biosecurity, the NSABB, only because of the anthrax attacks after 9/11, right? That was what prompted them to set up the NSABB. So in 2011, the NSABB deliberated and they put a moratorium on gain of function research. And the scientists voluntarily, stopped doing it and the papers weren't published, but that lasted only a few months. There was intense lobbying by the scientists, and, they were coming out with statements that were signed by thousands of scientists, including Nobel laureates, saying the future of humanity was at stake and we had to do this kind of research. But there were equally other scientists, including Donald Henderson, who was one of the US leaders of smallpox eradication, and Michael Osterholm, who's another well-known public health figure in the US, who came out warning about the danger of this research and saying, actually, the benefits don't outweigh the risk. We shouldn't be doing this kind of research. But unfortunately, the pressure and the vested interest was so powerful that those papers were published. So they're open access papers. Anyone can read up the methods and repeat the experiment, right, to create these gain of function engineered avian flu viruses. And after that, the floodgates were opened. There was research on creating a SARS version 2, right? Researchers were actually trying to engineer SARS-1 to make a SARS version 2. And a whole lot of other viruses were subject to this gain of function research. Then there was another moratorium on it because some people were still cautioning against it in 2014 that lasted till about 2017. In the meantime, the US and the European Union commissioned reports on the risk and benefit of gain of function research, but the reports they published were really very wishy-washy. They didn't come out strongly either way and didn't actually have any quantitative risk-benefit analysis, which can easily be done. So now it's, open slather. Anyone who wants to do gain of function research can.
Raina, you set me going. One more thing, if I may. Another recurrent theme throughout your book is the distinction between natural and unnatural viruses. And in the case of smallpox, which was long since eradicated, you refer at least once to the possibility of an unnatural smallpox reappearing. Can you say more about that rather appalling prospect?
Yeah, so if we suddenly see a smallpox epidemic in the world, you'd have to assume it's unnatural. There are people who say the melting Siberian permafrost could result in smallpox emerging from corpses buried in the Siberian tundra, but, odds are it's going to be a synthetic biology that someone's made it in a lab. Yeah.
Well, shall we turn now to questions from
Sure.
the audience? Please wait for the intern to reach you with the microphone.
Ian Thompson, the councillor. Question I have is looking towards, if you like, the positive side of it. I mean, one of the responses to COVID was the development of the mRNA vaccines, and other responses such as making sure everybody wore face masks and separated out, especially in the initial period. Is there a conscious effort to define those steps that respond to an outbreak so that when there is one, that they can be rolled out positively into the community as much as you can, and that there is immediate responses with vaccines and things like that? So are they doing proto-vaccine sort of development to respond to different viruses which may be not in any threat, but possibly if they're engineered or just circumstances arise that they become that?
Yeah, there are. Every country, almost all governments do pretty serious pandemic planning, and there's been a lot of efforts around vaccine development. It was really quite miraculous how fast we got the vaccines. The mRNA technology itself allows very rapid changing of the antigens in the vaccine, much more rapid, like in about 6 weeks you can change the composition of the vaccine, which the traditional vaccines take a lot longer, a year at least, right? So that's a really good thing, and we've now got 2 mRNA facilities set up in Australia, one in Melbourne, one in Queensland, in Brisbane. And that's going to be very beneficial to Australia to have that capacity. The thing about epidemics is they grow and we've got things like CEPI, the Coalition for Epidemic Preparedness, which played a major role in trying to get vaccine equity around the world and getting vaccine to where it was needed. And I think the problem, though, is that truly epidemic diseases and pandemic diseases grow exponentially. So something like malaria is an endemic disease. You might have very, very high numbers, but it's never going to crash your health system because it doesn't change rapidly over time. With a true epidemic disease, it grows exponentially, so you go from 0 to 100 in a matter of days or weeks. And that's why you see the bodies piling up in the refrigerator trucks. That's why you see people running out of oxygen in their health system, and so on and so forth. I mean, Australia planned very well for that aspect of it, and I think we expanded our ICU capacity by 120%. So we were really well prepared and didn't see those kind of effects, also by closing the borders and keeping it out until everyone was vaccinated, which was another benefit here. But that early stage of the epidemic growth, the drugs, vaccines, etc., come very late in the piece, relatively. Other interventions that could prevent pandemics entirely include detecting it very early, and because they grow exponentially, you want to pick up really early warning signals so you can actually go in and stamp it out before it spreads across international borders. And we've got something called EpiWatch, which is an AI-based observatory early warning system, which taps into open source data, so what people are talking about, what local news agencies are reporting on, and we apply the AI in 2 different subsystems to generate valid signals of what could be an epidemic earlier than any health department would know about it. So that can prompt earlier investigation, earlier diagnostics, earlier vaccine development. So I think we need to look at both ends of the picture, not just the pointy end where you're running around trying to develop the vaccine, but also in trying to prevent pandemics when they arise.
I've got a question, if I may. It all sounds like a pretty hopeless and gloomy situation going forward. And may I suggest that the more that governments and military and terrorists hear about and find out about how easy it is to develop these pandemics or whatever, the more danger we are going to be subject to. So whilst, of course, we need to know about it, and I'm one of these people who are finding out more about it from your fantastic talk this evening, by, how can I say, how do we go from here? I mean, it's the likelihood of something actually happening is quite high, and it's going to get higher over time. And who, early days of the pandemic for COVID, were not prepared to admit, as you mentioned earlier. So are they the right organisation for watching this or doing something about it? I mean, what is the solution?
So a couple of different points to unpick there. One is about, is it dangerous to tell people? No. I think the bad actors are well and truly on top of it. And no, it's like if you put a method for hacking a bank account online, it's 100% probability bad actors are going to use it, right? If you put methods out there on how to make pathogenic viruses more pathogenic, 100%, in my view, that bad actors will use it. They're onto it, they know the category of weapons they can use: explosives, chemical, radiation, nuclear, biological. I think it's always a catch-up game from the point of law enforcement, intelligence agencies. They're always playing catch-up, just as they are with cybersecurity, right? The bad actors seem to have access to better technology sooner, and the good actors have many bureaucratic obstacles and really slow wheels turning, etc. So I actually think one of the solutions, I don't think it's that hopeless. I think human beings are quite resilient and good at finding solutions. I think one of the solutions will come from more interdisciplinarity. I think the problem with, like, for example, looking at the origins of COVID is that in medicine and public health, we're never taught to ask the question, is this epidemic natural or unnatural? We think everything is natural. So that's a completely different discipline from police, for example, who are taught, you approach a problem in a particular way, you pull in different lines of evidence, it's like a homicide investigation, for example. I think it'd be great if early in their careers, when they're still training, that health people spent time working with other sectors like emergency response, defence, law enforcement, and vice versa. So you had people experiencing crisis emergency response from different perspectives, but also when you do that, when you create interdisciplinary training models, people build up lifelong networks and relationships, right, that are kind of organic and would help. Because when you see in these emergency situations, and I'm thinking about the West African Ebola epidemic in 2014, there was a lot of conflict on the ground between different responding agencies. Like there were defence people, there were military involved, there were emergency response, there were NGOs, there was WHO, and there was really no understanding or ability to work together harmoniously. And I think we could do a lot to improve cross-disciplinary training. If public health people understood, the way law enforcement people think about a question of origins of an epidemic, it would make a big difference. That's one thing. And the other is just raising community awareness, I guess. Because in democracies anyway, I think people do have a lot of power to change things. But I think, with this group that I've joined, Biosafety Now, they are trying to do some of the, trying to improve regulation of this kind of research. The pressure has to come on that side as well, to make it harder, to challenge, to sort of provide accountability for the people wanting to do this research. I think gain-of-function research and synthetic biology should be registered publicly, so that just like doing a clinical trial, you can't do it unless it's in a public register where everyone can go in and scrutinise exactly what you're doing, where and how, and that funding agencies, journals, etc., all cooperate to ensure that nobody can do it without complying with that regulation. So there's a lot of different things that can be done.
I'm going to be a bit greedy. I've got two questions I'd like to ask you. The first one is, I'd like to ask you why the development of anthrax by the UK in, I think, the 50s and 60s. They went to that island in Scotland and they killed thousands of sheep developing anthrax, which was well known at the time, publicly, and they didn't attempt to hide that as far as I understand. Now, admittedly, there wasn't a breakout of anthrax, but it was well known that this was a deliberate, biologically engineered weapon by the UK government. And compare that to the reaction when COVID broke out and the implication that it may have come from the Wuhan lab. There seemed to be enormous world political pushback against that as a suggestion. And the WHO delayed making an investigation for 1 year when, of course, they could scrub from top to bottom the entire building and get rid of all the evidence, right? Why was there such an enormous difference, that there's a revulsion that the concept that this may have come from a lab, when it's well known that around the world for decades biological weapons have been developed, including nuclear bombs, and nobody, you can hardly hide a nuclear bomb. But why the revulsion against China to even suggest that this could have come from a lab? That's question number one. Number two, you mentioned Ebola very quickly. When that came out, there was an enormous panic about how contagious it was, and they showed these people dressed from top to bottom in plastic tents and scrubbing everything. And from one day to the next, Ebola just seemed to disappear, and there didn't seem to be any inoculation program. So how could that have gone from being such a horrifying pandemic, or mini-pandemic, I don't know what you call that, to self-extinguishing through what seemed to be no direct human intervention compared to COVID? So sorry, there are two questions, but...
So the first question, I think, is just the era. The Scottish island where they were blowing up, animals with anthrax, was during the Cold War. It was completely different era when there was an arms race in biological weapons, overt. So the US was doing similar experiments, as was the UK, and in the book, my opening chapter is actually something called Operation Sea Spray, which is where the US Navy sprayed bacteria over San Francisco to see how far they could aerosolize these pathogens. They didn't think it was a harmful bacteria, but actually people did get sick and one person died. It was declassified about 30 years later, when the son of the man who died kind of pushed, and then a journalist uncovered it, and eventually it was declassified. But there was lots of crazy stuff going on, like they were dropping light bulbs in the subway in the UK and in the US, filled with trillions of spores of bacteria to see how far they'd disseminate with the movement of the trains in the tunnels. So they were doing all kinds of experiments. But I think in the Cold War, it was a different kind of era. And you could argue that the same sort of stuff is still going on, but it's much more covert. Yeah.
And the Ebola story?
So Ebola has a reproductive number of about 2, which means one case will on average give rise to 2 additional cases, compared to the current variants of COVID, where it's more like 8 to 10, right? So super infectious. So that is kind of like similar to influenza. That's the kind of range of infectiousness of influenza, but it's not not predominantly a respiratory pathogen. It's spread through contact with blood and bodily fluids. And initially, the epidemic grew so large because, and this was actually the epidemic that inspired me to start developing the EpiWatch Observatory, is there was a very long delay. So the WHO knew about it in March, and there wasn't any concerted international response till August or September. So in that time, the size of the epidemic grew in a few months from a couple of hundred to like 20,000 cases. So, and at that point, we saw the stress on the health system where the hospitals couldn't cope. And this, West Africa is one of the poorest regions in the world, and rich in natural resources, but the people don't have those resources. And So people with Ebola were dying on the streets. They couldn't get to a hospital. And everyone was just, panicking in a flap, not knowing what to do. All the global health people, etc., they were writing lots of papers, publishing papers in prestigious articles describing, the death toll and what was happening, but no one had any solutions. And then one American group published a modelling paper showing that if you could get 70% of patients into a hospital bed, you could stop the epidemic. That the contagion was going on because people were dying in the streets, that there wasn't anywhere to isolate them. So at that point, there was like a field response with militaries and so on, and other agencies responding and setting up tent hospitals, field hospitals, etc. So they expanded the capacity to isolate patients and treat them, and that brought the epidemic under control. So there's a lot you can do just with non-pharmaceutical interventions to control an epidemic.
Hi, Raina. My question is about further up the pipeline, so your suggestion around publicly registering dual use, not dual, but gain of function and synthetic biology. Given that there's a competitive advantage to developing synthetic biology, how would you go about getting global cooperation on
the same
public?
argument could be applied for clinical trials, right? It's all competitive, all of the research, whether you're testing a new drug or whatever. So I think it's possible, As I said, that was a massive shift. And it was enforced, and it's now nobody would even dream of, well, I'm sure some people do it, but they wouldn't get it published in a reputable journal, or they wouldn't get funding from reputable sources.
Even in this geopolitical climate, would you say, given, you could have warfare implications?
It's already public. I mean, now there's been a huge move in the last 15 years for researchers to make everything open access. 15 years ago, a lot of the research was behind a paywall. Now, researchers have to bear the cost of making their research open access. So if I get a paper accepted in a reputable journal, generally speaking, there's a few that don't charge. They will charge you page fees. It's usually in the vicinity of $3,000 to $5,000 per paper, right? And so once the paper's gone through peer review and it's accepted, so that means it's then available for anyone who wants to read it. So there's nothing really to hide because it's all available. And you don't have to necessarily put down all your methods. You can still declare what it is you're doing and your basic objectives and so on. It's like there's a lot of other models for that kind of transparency. It's like the American Immunization Committee, ACIP. All their deliberations are done in public. So people can watch live streamed the ACIP deliberations, which is different to here, where when a TAGI deliberates, no one knows what's gone on. The minutes aren't available. Even in the UK, the minutes are available of the meetings. But in the US, it's completely open. Anyone can go and listen or watch it on live streamed, and it's completely transparent. So I think, that's a step in the right direction.
Thank you, John Murray. You mentioned that some of the superpowers are developing human beings who would be then programmed to join their armed forces. Could you elaborate on how that action would be taken?
So I guess the objectives with trying to engineer people to be super soldiers is, being resistant to pain, having increased stamina, being able to heal faster, being stronger, fitter, all of those things. That will be a combat advantage. And it's happening. Definitely in the US and the UK, probably in other superpowers as well.
How is it disseminated into the population?
What do you mean?
Well, once you've done that genetic engineering, do you then inject that into something, or?
There's different ways you can confer changes to the human genome. It can be done through ingesting something, for example, but I think it's pretty likely that in the next 10 years, people will be able to choose the genetic characteristics of their children. That'll become routine. Probably it'll be accessible to wealthy people first, but it'll become more and more accessible. That's what I think. And it's got pluses too, because, you know, you can get rid of genetic diseases that way. You know, people who've suffered because of cystic fibrosis or other rare genetic diseases, that can be treated, cured even through this technology. But I think it will expand to other uses, you know, where parents will want, you know, will be, that's what I see happening in the next 10 years. A bit like the movie Gattaca, right? Seemed like complete sci-fi back then, right? But it's a reality.
Thanks for your talk. Gain of function research sounds pretty scary, but you mentioned that there are arguments about it in terms of some people claiming pros as opposed to the cons. So what arguments do people make for gain of function research, and then what is the motivation to do it?
That it's helpful to develop drugs and vaccines, or to predict the next pandemic pathogen. So you make the pathogen to predict that that's what's going to cause the pathogen. There's actually no concrete examples of where it's been beneficial. Yeah.
And so what advantage do commercial or other interests get from it?
I don't think there are that many advantages, and there's other methods you can use to sort of do the same kind of research. There's a technology called gene silencing, which you can also use, which doesn't result in permanent genetic changes to living things. And you know, a good example is the 2009 influenza pandemic, if you remember, the swine flu. Leading up to that, from 2005 onwards, there was very intense pandemic planning, and people made precursor vaccines. I think you asked about the precursor vaccines for H5, the hemagglutinin antigen of the H5N1 avian influenza. And, you know, with influenza, you need an exact match to the virus to give you protection. Partial matches might give you a little bit of protection, but it's really not going to cut the mustard. So they developed these H5 candidate vaccines that would give you some, you know, partial, minimal protection. But of course, the virus that emerged in 2009 was H1N1, completely different virus. So those vaccines were useless. You know, if it is truly a natural pandemic, you can't predict what the virus will be. And, you know, going to remote caves where no human beings even go and bringing back dangerous viruses, thinking you're going to, you know, find out what's going to cause the next pandemic, well, it's probably going to come from the lab that you took it to.
Just to follow up a bit on that question, obviously the people engaging in that research, and it seems to have been the Chinese with some American involvement at some stages, and obviously they saw it as having advantages. They had their reasons for doing it. There's many people in many countries doing that kind of research. As I said, it's an industry. You know, these are careers, people's careers. They've built their careers on it, and if you're telling someone to stop doing something on which their career depends, of course there's going to be pushback.
Oh, thanks. Geoffrey Hawker, Politics Macquarie. Thanks very much for your talk. I'll be interested to read the book. I've already got the main message, I think, which is that people like us should know what's going on. To me, that bounces off two issues: national governments and the pharmaceutical industry broadly considered. Now, I think the reason why WHO can do this but it can't do that is, of course, it's constrained by the power of national governments, certain governments in particular. As I'd understand it, because of nationalism, we see it all over the world. To put it briefly and crudely, there's hardly a national government anywhere that doesn't have an interest in watching its back, outstripping its rivals, and is totally against any form of regulation unless they're forced to it. they're hand in glove with the pharmaceutical, broadly considered, industry that won't be regulated either. It's not just what you've been talking about tonight, it's across the globe with chemicalisation and all the rest of it. So of course we, the people, are at all times fighting contained power in the public and the private sector. That's the condition of our life. And I think what you've been talking about tonight simply exemplifies that vividly as you've done. I just wonder if I can make it a little bit more concrete and pose it as a question, because I think everything I've said so far is pretty straightforward, actually. What about Wuhan and the American involvement? Can you, I've been reading about this, but can you explain, please, a little bit more why the dreaded Chinese and, the dreaded Americans are there together, and what the consequences of that sort of cooperation, hostility cooperation, joint thing between them is? I'd be really interested to know that.
So first of all, research is very international, and international collaborations are really common, and they're a good thing in general. But the second thing is certain settings may allow research that wouldn't be allowed in other settings. So, and I'm not naming countries or specific circumstances, but there are other reasons why certain research partnerships may occur to enable research that couldn't be done just take my face mask trials. This is another, it's an example. We tried to do face mask trials in healthcare workers in Australia and nobody would wear a mask. So we realised we can't do it in Australia. So, I formed a collaboration in China and we ran those trials in China because healthcare workers will wear a mask there. So that's just a very sort of innocuous example, but I think when it comes to gain of function research and so on, there could be other issues that would not be allowed to progress beyond a certain point in one country, but may in another.
We have time for at least one more question.
Chris Skinner. Sorry, was there another? I'm interested to know to what degree artificial intelligence is being applied to the research that you've been describing.
Yes, very important. AI has been applied everywhere across medical research in all different ways. For example, 20 years ago, to develop a new drug, you'd have to go through painstaking experiments in animals over and over again, sometimes for years. Now you can do it computationally with AI, knowing the sort of chemical makeup of the drug and the makeup of the cell, the target cell. You can actually do it computationally in very short periods of time and cut out a huge amount of experimental research. So it's very important, AI, in research.